immunoglobulins or antibodies. You can see in this diagram, this is a Y-shaped protein. Y-shaped proteins. Y-shaped glycoproteins. These are the glycoproteins. The immunoglobulins are the glycoproteins and composed of the two identical chains. H chain means heavy chain, H chain, heavy chain, and light chains L chain. Heavy chain, H chain, heavy chain, and light chains. These chains are covalently held together. These chains are held together by disulfide bond. You can see it's disulfide bonds. Disulfide bonds creating a bilaterally symmetrical structure that can be categorized into the distinct functional structural regions. Distinct functional regions. The first thing is these arm of the Y-shaped molecule. There are two regions, variable regions and constant region. You can see it, variable regions and constant regions. So variable regions, the tip of the arms forms the variable domains. The tip of the arms, these are the two arms YK. This is known as variable regions. Variable regions which are made up of the one heavy and one light chain. One heavy and one light chain. ये short light chain and ये heavy chains. So one heavy and one light chains. This region contains hypervariable amino acids sequence known as complementarily determining regions, and that creates the antigen binding sites. So this is the variable regions. Then constant region below the variable tip, this structure is known as the constant region. And FC, what is FC factors? Crystallizable factors. What is FC fragment? FC fragment क्या होता है? FC crystallizable fragment. This forms the stem or base of this Y-shaped structure. This is known as crystallizable fragment, FC fragment, and this is constant region, and the base of the heavy chain determine the class of immunoglobulins. जो ये base है, that determine the class of the immunoglobulins. And this region is responsible for binding. This region is responsible for binding the specific cell receptors, FC receptors, and the activating the complement system. The next is the hinge region, so found between the Fab and FC region. This is the hinge regions. And light chain types, all immunoglobulins contain either kappa particles or lambda particles. I am repeating again, all immunoglobulins contain either kappa particle or lambda particles. So based on the heavy chains structure, immunoglobulins are divided into 5 primary isotypes. I am repeating again this important thing. Based on the heavy chain structure, immunoglobulins are divided into 5 primary types: IgG, IgM, IgA, IgD, and IgE antibodies. And we will discuss the functions of these antibodies in our next lecture.